going to get a little practice with p-values and t-values and that kind of thing. So we need a data set. And a kind of a fun data set is to look at the genome of Saccharomyces cerevisiae, that is to say, baker's yeast. So here's a little picture of some yeast cells growing in a cell medium. Here's a little diagram showing some of the things inside. Yeast is a single-celled organism, but it's a eukaryote, just as humans are. In fact, in the great scheme of things, yeast and human are actually pretty close. This is a tree of life drawn to scale by the evolutionary distance of different organisms. And you can see that human beings are here and yeast is here, all in the eukaryotes. And then quite distant are all the bacteria and all the archaea, which are things that you might loosely call bacteria, but you can see from this tree of life they're really quite far from being bacteria. Now what distinguishes the eukaryotes, the things on this branch, are that they have chromosomes. Humans have chromosomes and yeast has chromosomes. And we're going to take as our data set, a very simple data set, it's simply a very long string of A's, C's, T's, and G's. Those are the bases in the genome. And we're going to use the string which is chromosome 4 in yeast. It's about 1.53 million letters long. And it begins ACA, CCA, and so on. Then I've omitted 1531894 letters, and it ends TAGC, TTTT, GG. There's a text file on the course website that you can download that simply contains this very long string of letters A, C, T, and G. And to get a little bit of practice with something very simple, we're just going to count them all. If you count them all, and I hope you can all very easily write a program to take a text file and count the frequency of occurrence of the letters in it, if you count them, you'll find that there are this many A's. And let me go out of order and show you that there are this many T's and that those numbers are nearly the same, but not exactly the same. And there are this many C's, 289,341, and a very similar number of G's, but again, not exactly the same. So, as null hypotheses, let's ask a couple of questions. The first null hypothesis we might have is, are these counts consistent with a model that A, C, G, and T is randomly chosen with probability 1/4 in each position? Well, you might guess from these numbers that that can't possibly be true, but how do we know? What's the formalism behind that? What is our degree of certainty that a random model could not produce these numbers? We'll answer that question. A much more interesting question, and it's also even interesting biologically, is could we take as the null hypothesis the hypothesis that the probability of an A in each position is identical to the probability of a T in each position? And I've done the arithmetic of dividing each of these by the total, and so we would have a hypothesis that says P of A equals P of T is equal to some number that we won't try to pin down right this second, 0.31 approximately, and similarly that P of C is equal to P of T is equal to some number which seems to be about 0.19. Well, you might think that that is a consistent model because of the way DNA works. In DNA, every A base pair is paired with a T. I should say A and T make a base pair. And every C base is paired with a G. And it kind of looks from this picture of the way the strands work that there should be exactly the same number of A's and T's, exactly the same number of C's and G's. How can that be that they're not? Well, I'll give you a hint. What's in the text file here is one strand of the double-stranded helix. And it's consistently the same strand all the way down through chromosome 4. So if these numbers turn out to actually be statistically different, it will mean that there's a statistical difference between the two strands of chromosome 4. Whereas if these two numbers turn out to be close enough that they're consistent with having equal probabilities, and it's just random fluctuations that are making the counts different, then we might think that the two strands should be statistically identical. And that's the biology question that we'll be answering in just a minute here. So as always, the starting point is to write down a model. If we're a Bayesian, we write down a model for what is the probability of the data. But now we're being frequentists, and a frequentist writes down the model, what is the probability of a test statistic for the null hypothesis. A possible model in this case could be multinomial. That is to say, at each position in the approximately 1.53 million positions in that string, we would pick an independent and identically distributed choice of A, C, G, or T. And so we would have four numbers, probability of A, C, G, and T, adding up to 1, and those four numbers would be the multinomial model. Now that's a perfectly good model. In fact, it's probably a better model very slightly than the one we're actually going to compute, but we're going to compute one that's a bit simpler and almost equivalent. And that is, we're going to compute four separate binomial models. In other words, at each position, independent and identically distributed, we're going to ask, is it an A or is it something else? And we're going to use that to define a probability P of A. Then we're going to separately ask for each position, is it a C, is it a G, and is it a T, and see what we come up with those four separate binomial models. The difference between the four separate binomial models and the multinomial single model, if you think about it, is that in this case, we're not constraining the four separate fluctuations to add and subtract out to the total number of bases in the string. In other words, in real life, if there are more bases than there should be by a statistical fluctuation, more A's than there should be, then there would have to be on average fewer C's, G's, and T's. And we're not going to account for that correlation between the probabilities in our four separate binomial models. But otherwise, this is a perfectly reasonable model to use for estimating the probabilities A, C, G, and T. Now the model is binomial, but the counts are all so large here that we can approximate the binomial by a normal distribution. So the way that works is that a binomial distribution of making n draws of something, each with probability p, is almost the same distribution for the number of draws that turn out positive as the normal distribution with mean n times p, and with standard deviation, what's given by this term over here. Where did this come from? This is actually a trivial relationship for us to prove because we know the central limit theorem, and we know that the binomial distribution is what you get from Bernoulli trials. So, in fact, the binomial is a sum of Bernoulli random variables. In other words, we're going to do n tries of a random variable that either comes out 1 with probability p or 0 with probability 1 minus p. And then we're going to simply add together all these values, and therefore adding together all the values of 1 will simply be the number of successes we had out of n, which is exactly the binomial distribution. Now for a single try of a Bernoulli variable, we know how to compute the mean and the standard deviation, so let's just do that here. The mean is the first moment, so that says with probability p, we get the value 1, and with probability 1 minus p, we get the value 0, and we add up the two cases, and just staring at the arithmetic, it's obviously p. So the mean of a single Bernoulli try is simply p. And what is the variance, or standard deviation squared? Well, that's the centered second moment of the distribution. That is to say, with probability p, we get a value 1, and its distance from the mean squared is 1 minus the mean squared. Here's the value of 1 that we got. And with probability 1 minus p, we're going to get the value of 0 shown here. So its distance from the mean is going to be 0 minus mu squared. I've written it out that way for clarity. And now if you just do the arithmetic here and substitute in mu equals p, you'll see that this is p times 1 minus p. Now the central limit theorem. The central limit theorem says that if we add n such random variables, I guess I'm a little confused in this slide between capital N here and little n here. They're both supposed to be the same thing. So if we add n random variables, the mean is n times the individual mean, or np, and the variance is n times the individual variance, or n times p times 1 minus p, and of course the standard deviation is simply the square root of the variance. Okay, let's try this out to see if we can rule out the silly hypothesis that all the probabilities are equal, and since there are 4 of them, they must be equal to 0.25. Now we need a test statistic. Our test statistic is going to be the value of the observed count under the null hypothesis that it is binomially distributed with probability p equals 0.25. And we saw that because the counts are so large, this is effectively the same as the hypothesis that it's normally distributed. So let's put in the numbers. The mean that we expect is 1/4 times the total number of base pairs. That is the 1.53 million. The standard deviation we expect is p times 1 minus p times the number of base pairs in the whole string. And to get standard deviation from variance, we take the square root. Now here we'll compute a t-value to see how many standard deviations away from the mean is what we observe. And that would therefore be the observed count minus the mean that we just computed divided by the standard deviation. So far that's just arithmetic. The slightly harder part is to convert the t-value to a p-value. That is to say, how extreme measured by area under the curve is the observation that we observe here. So we'll go over to this picture here. Here's a little sketch of a normal distribution. Now what we can look up in MATLAB or numerical recipes or any library is the function which is the cumulative distribution function, capital P, of the normal distribution. And that's defined as being the area under the normal curve starting at minus infinity and going up to some point t measured in standard deviations. So if we want the area of the right-hand shaded region, for example, if the t-value were right here, that would be 1 minus the cumulative distribution function at that value of t. And here I'm giving the example for positive t, so it doesn't matter if I put in absolute value of t. And here I have that 1 minus the cumulative distribution function up to the value t. Now, why have I also shaded this left-hand piece here? Because the way a p-value test works is we want to know the probability of seeing data as extreme or more extreme than what we actually observe. And we have to make a decision about what we mean by extreme. We've discussed this before. If we have way more counts than we expect under this model, then we would be in the area of the curve on this side. But we would also consider the null hypothesis ruled out if there were way less counts than we expect under the null hypothesis, and that would be an area of the curve on this side. Now, by the symmetry of this picture, it's the same area as long as we evaluate this cumulative distribution function with a positive number. That's why we have the absolute value signs. And the fact that we're including both tails, that it's a two-tailed test, is why the p-value gets this extra factor of 2 right here. Well, now let's just put in the numbers and find out what it comes out. I'm not going to show you the actual computation, but I'll tell you the result. The t-values come out these extraordinarily large or large negative values here. And when you put these t-values into the cumulative distribution, you essentially get 1 for this and 0 for this. In fact, it underflows on the machine when I tried it. And so the p-value is effectively 0. This really, really rules out the null hypothesis that the probabilities are equal and are equal to 0.25. So let's try a null hypothesis that might not be ruled out, that might be a reasonable hypothesis. And as we saw already, the counts of A's and T's seem suspiciously similar, and the counts of C's and G's seem suspiciously similar. So let's take as the null hypothesis that A and T occur with identical probabilities, and that C and G occur with some different identical probability. As our test statistic, we can take the difference between the counts of A and T, and separately when we do CG, the difference between the counts of C and G under the null hypothesis that they have the same P. What I mean is that A and T have the same P, or C and G have the same P. Now we don't know when we start what the value of P should be. In the previous model, we knew by symmetry that it had to be 1/4. But here it's a parameter in the model. And frequentists estimate parameters typically by maximum likelihood estimates, and we're going to do that here. Go back to the lecture on Bernoulli trials to see what the maximum likelihood estimate of a probability P is in this case of binomially IID distributed values. So here are those maximum likelihood estimates. It's basically that if we want to know the probability of A or T, so we group together the counts of A and T, and we divide by n. That's the maximum likelihood estimate of the probability for A or T. And now, since the null hypothesis is that they're equal, they would each be equal to half of this, so we have the half in front. And similarly, a formula that estimates the probability of a C or G in the data. Now we've got the parameters, so we need to compute the test statistic. Well, let's see. Our model is that the counts for A are drawn from a normal distribution. And the parameters of that normal distribution, its mean will be the total number of base pairs times the probability of an A. And the standard deviation, as we saw before, will be that probability times 1 minus that probability times capital N, and then the square root. Similarly, the probability for T will be exactly this same expression, because under the null hypothesis, the probability of A and the probability of T are the same. Now the test statistic is the difference between these two counts. So to do the p-value test, we have to compute the probability distribution of this quantity, the difference between n sub A, the number of A's, and n sub T, the number of T's. Here, because we've made the approximation of a normal distribution, it's very easy to know what this distribution is, because the difference of any two normals is itself normal, and it's only a question of figuring out what the mean and standard deviation are. Well, since NA and NT have the same mean, here it is, the mean of the difference must be 0, so we can put a 0 here. And then slightly more subtly, the difference between two normals has a variance which is the sum of the variances. That's essentially the same theorem that the sum of two normals has a variance which is the sum of their individual variances, because the variance doesn't really care whether the quantity is a positive quantity or that we add, or in effect, negative quantity that we add to do the subtraction here. So therefore, the variance is twice the variance shown up here. That's the factor 2 here. And we now know what is the distribution of the test statistic. So all that's left is to put in the numbers of the actual data and see what the p-value is for that test statistic. Now I have to make a little aside here as a Bayesian, because although we're being frequentists for a while, we're still deep in our heart Bayesians. And it makes Bayesians a little bit nervous to see the procedure that we're adopting here. The procedure where we first compute estimates of the parameters of the model and then do something for goodness of fit to decide whether it's a good model or not, whether we can rule out the null hypothesis. And the reason that this makes Bayesians nervous is that the data is kind of being used twice. The data is first being used to estimate parameters and then being used to estimate goodness of fit. And you kind of wonder whether that's fair. Bayesians would feel much more secure in estimating a full posterior probability of all the parameters at once. For example, these probabilities and this difference and so on. We'll come back to this a little later when we talk about Bayesian criticisms of p-value tests. But to put in the numbers for this model, here I've calculated it in MATLAB. So we start off with a vector that has the two differences we're interested in, the differences between A and T, and the differences between C and G in their counts. And then we saw how to make the maximum likelihood estimates, and here are those maximum likelihood estimates for the probability of A or of T, and the probability of C or of G. And we're going to do normal distributions, and they'll have mean 0 as we saw, and standard deviation, and then here's that formula where the 2 here comes from the fact that it's the difference between two normals, each of which has this variance. So the t-values are here. Notice that in MATLAB I'm computing on the vectors, so I'm computing the two values, the AT and the CG, both at the same time in these formulas. And then the p-values, well, I need the cumulative distribution function of the normal distribution, and in MATLAB that's called norm CDF, and go look at the MATLAB help file if you want to know why I have to put 0, 1 here. It should be obvious. It's mean 0 and variance 1 for the normal distribution. Anyway, MATLAB does the arithmetic, and look what we find. Surprise! We find out that the t-values for both the AT and CG cases are actually pretty large, in this case large and negative, and the p-values are therefore quite small, both of them less than 1%. So this model is actually ruled out with high significance, that is to say with small p-values. And it's just not true that statistically there are the same number of A's and T's in the yeast genome chromosome number 4, or the same number of C's and G's. That might surprise you, understanding the underlying biology, but actually this is underlying biology. What's going on here is that we've discovered genes. Let me explain to you why that is. Our hypothesis was that each base pair was independently random on, for example, the plus strand of the double helix DNA. But that's actually not correct. The reason is a whole sequence of base pairs form a single gene. So this might be one gene, this might be the next, this might be the next. And genes actually have a preference for biological reasons. It could be that this gene tends to be more A-rich and less T-rich, or that this gene tends to be more G-rich and less C-rich. So our hypothesis of independently random base pairs along a strand is just not true. The reason the counts are therefore not as close as they ought to be is because the number of genes, which would be kind of the n in those binomial formulas, is much smaller than the number of base pairs. So the fluctuations that you might expect are correspondingly larger, because they're fluctuations not of how many base pairs are A versus T, but of how many genes are A-rich versus T-rich. We could actually put numbers in this and try to estimate quantities like how many genes there are or what's their average length. We could actually estimate a certain combination of those two. But that goes beyond what we want to do in this unit.